So every week, the first thing we do is take our quiz on the previous lab, right? So you just took the quiz on lab 1, which we did last week. This week, we're going to do lab 2, right? So guess what the quiz is on? Lab 2. What we covered today is on the quiz next week, right? What homework are you going to do? Lab 3, right? And as I said, if you miss lab ever, you can go pick it up from my office door because I print enough copies for the number of students we have currently enrolled. So if you're not here, there's a copy on my door for you. If you're in Andrew or Sarah or my section, everybody but Kendra. Or you can go to Canvas and it should be posted on Canvas as well. So here it is on Canvas. Your homework for next time is not so bad, right? It's 2 exercises, a little bit of terminology, and then I want you to watch some videos that are here on Canvas. The next pages are actually what you're going to do in lab, and you're going to fill in lab. But I kind of pre-wrote out for you guys so you guys get an idea of the kind of prep work you'll need to do in the future. So you'll fill in, you know, what organisms grew, right? Once the ones we picked, right, are ready, we'll know. And then you guys will do this procedure. So this is kind of fill in the blank. You'll do that in class. But read over it ahead of time so you have an idea of what we're doing. And you're going to read the exercise first, of course, too. And then do your terms, watch the videos, read over this handout. And so for this first one, I wrote out the procedure for you and even a checklist. But in the future, you guys are going to start writing out your own procedures ahead of time, right, and preparing for lab. So for this one, and as I'll do today, as I found out was a good thing to do, I have recorded my demos, even though I'm going to still demo next week. I recorded them, and I have them posted for you here under this module. Andrew has them too. He should post them for you guys. These are the videos, YouTube videos, so of me demonstrating, and in fact, this one I make a mistake. And I want you guys to write on your sheet what mistake I made. So you gotta read over the protocol, right? Read over the sheet, and then watch this video and see if you can figure out what step I skipped. Okay? So I'm going to be asking that next week. Right? Who knows? It might even be the bonus question on the quiz. And then there's some other videos here that are other YouTube videos that are good ones. But definitely watch the ones of me, because that's what we're going to do. Not that I want to be a YouTube star or anything, right? It's not my goal at all. But I do this to help you guys, because the more you see things, right, and the better you can see things, the smoother and more successful you're going to be in lab. So, I already did attendance. So the other thing is the book, and it was so exciting to see books in class. I can't even tell you. So they're in. So go get yours after class, right? Be one of the first 50 that gets a book, because they only ordered 50, and there are currently 66 students enrolled. And Yolanda's like, tell them it's not my mistake. I'm like, no, you still made a mistake. You just don't want to admit it. Right? That you're going to be ordering, and there's still going to be students waiting on books, right? Because she still hasn't, as of right now, ordered more books. So reserve your copy, go get your copy today, right? Do whatever you need. If for some reason this first batch you're not one of the first 50 that gets a book, you need to show me a receipt that you've reserved on their website. They'll send you an email. You need to email that to me, and then I will email you the next lab of the lab manual. I will not give it to you without showing me that you are trying to get it. Does that make sense to you guys? So lab 3 is already posted here. I'm new to scanning. I did this myself. So one of the pages is out of order, but they're all there. I don't know how to fix it other than to start all over again, and I was frustrated as it was on Tuesday. Okay? The no internet thing made me very unhappy, and I broke my microphone. It's taped. So, just roll with it, right? We're rolling with it. You guys are doing a good job rolling with it. Thank you for that. So, in addition, like today when I do the lecture, and you'll notice I already have the lecture posted for you, And then after lecture, I'll post the link right here of the recording that I'm doing right now. So you'll have access to that. I'm also going to have these handouts that we'll hand out with the slide trays. It's pictures with the arrows pointing to the things you're supposed to identify. So these were really handy. Instead of having, even though you guys do have lab manuals, instead of having them out and flipping through them, these are quick one-sheet guides. There's 2 per lab bench. They're different from each other. They have all the organisms that you need to find under the microscope, so they help you reference, make sure you're seeing what you're supposed to be seeing, and help you identify and find what you're supposed to be identifying. So your key right here, although I don't know why Peter highlighted the mold. I guess he was, it's the scientists in us. We try to group and categorize things. But the really important thing on these is the name of the organism, right? Aspergillus, which is like right in the middle. It's going to be italicized though, right? Because it's a proper name. So that's going to be the name you see on the slides, right? Each one of these trays has all the slides you need. So you share between you and your lab partner. One set, one of the really expensive slides, I didn't order that many of because they're really expensive. So you'll have to share that one slide amongst the whole lab bench for 3 of the trays. And it says it on the tray. So when we actually get to looking at these guys, the unknown mold is over there, right? And so that is literally from an auger plate last semester. I cut it, and I took that slice, and I put it on a microscope slide, and I stained it, and I cover slipped it, and I sealed it, so that we could look and see at the microscopic level the hyphae that make up mold, those long filaments. So you should have seen these whole bunch of like blue lines, right, under the microscope. So at some point, if you haven't looked at that microscope that's on over there, make sure you do so. And there's even the label in front of it so you know what it is. So those pictures right there, so you guys aren't taking pictures of my pictures, I posted them for you. I just didn't do that ahead of time because remember, for homework you were supposed to, if you read right here, for homework, you were to complete lab 1, do the terms, right, and take the quiz. That's a practice quiz. It doesn't count for points. You can still do it if you want to. It'll help you study. And you were to complete lab 2 handout and to draw the organisms based on the pictures in your lab manual on the eukaryotic ID sheet. And then here is the lab manual even for you to help you do that. And then, of course, study for the quiz 1, which hopefully you guys did do that. Right? And this was the link to the lecture, and then the recording was there as well. I also wanted you to watch this video ahead of time, but I'm also going to play it in class. This goes over how to focus your microscope. Right? It's much easier for you guys to see it up on the screen. That way you can all see what I'm pointing to on the microscope instead of me doing it at the front of the room. This is a YouTube that I did with a colleague of mine. The audio-visual department came in and did another video, a nice professional one, except the editor screwed up. And so they'll be filming me again this semester. And so hopefully I'll have an even better one that's going to be distributed to all the biology labs so they can all project it up on the screen and everybody can see what you're supposed to do with a microscope. Not really, but somebody's got to do it. Okay. It's my service project to the department. We're actually required to do service projects. So, this is one I think is worth my time and effort, right? Teach people how to properly use microscopes. They're my favorite toy. All right, so any questions on that? All right, we understand what we're doing and what we need to do. Okay, so let's go ahead and get into the fun stuff. Let's actually talk about what we're going to look at today. And so this follows right along with your Lab 2 handout. I didn't open it already. Where did it go? Lab 2. So it's just one exercise, but if you've read over it or looked at it, right, it's a lot of stuff, right, a lot of stuff. And this is the only lab that we're going to deal with eukaryotes. So definitely you guys in my lecture lab should know what's a defining characteristic for a eukaryotic organism. A nucleus. So for most of these guys, one of the things you're going to need to find and identify is, guess what? The nucleus, right? And these are going to be relatively large cells as compared to bacteria, which are what? Eukaryotic or prokaryotic? Prokaryotes. They don't have a nucleus. They're much smaller. So quite a bit of terminology. So we kind of did a glossary of terms in the front of this PowerPoint for you. I'm not going to read this verbatim. You can read it. You have access to it now. But we're going to talk about each one of these throughout the lecture. But this is a good, glossary for you for the terminology. So again, we have two cell types that we talk about when we talk about cells. We have prokaryotic, which lack a nucleus. Eukaryote, which actually means true kernel. Right? So that nucleus in the center looked like a kernel inside the cell. And so that's how they got their name. So for this picture here, you have a scraping that was taken from the gum line of an individual, and it was stained using the Gram stain. And you can see that the epithelial, and those of you guys in A and P, squamous epithelial cell. Remember that? Right? They look like fried eggs. They're spread out. And here's the nucleus, right? And there's the cytoplasm. And you can see the border, the membrane of that cell. That's a eukaryotic cell. It's very large, right? Has a very clearly defined nucleus. Here's another one right here. It's probably underneath this one even, right? They're overlapping each other. And then you see all these little purple things. These are gram-positive bacteria. Much smaller, no nucleus. So the good news today is the organism we're looking at are relatively large, and most of them you're going to see a nucleus because they're all eukaryotic organisms, right? No bacteria today. After today, we will live in the bacterial world, right? All prokaryotic, all bacteria, all teeny tiny. So you're going to learn a technique today, and we're going to be using it from now on, and that's the use of oil immersion. All the way to the oil immersion lens, which you probably have never used in any of your other classes, right? But in our class, we can't get away without using it. We have to do it to be able to visualize bacteria because they're that tiny, right? We got to go all the way to oil immersion. So this classification system, right, of grouping organisms has been done since Carl Linnaeus back in 1758. Right? He started grouping organisms and classifying organisms. This study of classifying organisms is referred to as taxonomy. And of course he published a manual, right? And initially organisms were classified as plants or animals. We know that's not it nowadays, right? We have things like bacteria. Is that a plant or an animal? Well, it's really not either, right? It's its own classification. So classification, too, has changed over time as we've discovered new organisms, right, and discovered more things about the organisms in which we know about. So it has diverged quite a bit, even from this schematic that you see here that most of us learned when we were in school or in biology class, right? We learned about kingdoms and phylums and classes and orders and families and genuses and species, right? So like this diagram here, right? You have the animal kingdom, and then you could go down into a smaller grouping referred to as the phylum Chordata. All of these have a unifying characteristic. Anyone know what the unifying characteristic of Chordata is? Cord. Spinal cord. Yeah, backbone. Right? A spinal cord. And then when you get to the class Mammalia, all of these have unifying characteristics of mammals. Anyone know what some of those are? Hair. Warm-blooded. Warm-blooded. Mammals. Mammals. Mammary glands. They all have mammary glands that produce milk. They give birth, right? Live birth for the most part. Okay? These are unifying characteristic of mammals. And then when we get down to carnivores, what do they all have in common? They just eat meat, right? Although not necessarily true for bears, right? They eat berries, right? But There are other characteristics, more definitive characteristics for carnivore. I don't have them memorized, right? And then you get into the bear family, right? They all kind of look the same, right? So again, they have unifying characteristics. And then we get down to the genus level and then to a single species, right? And when we say a species, we actually include the genus name, right? So it's always the genus and species name. The genus name is always capitalized. The species name never is. Right, it's never capitalized. When we're writing them, we underline them. When we're typing them, we italicize them. So notice that it's italicized for the proper name, species name of an organism. So you'll notice one classification above kingdom here, called domain, right? And that is a newer system. So how are these groups determined? What do we talk about when we said phylum and mammals? Characteristics. But nowadays, they also take into account ribosomal RNA sequence. And this is actually how we came up with the 3-domain system. Above kingdoms was based on the ribosomal RNA sequences. So remember, what domain are we looking at today? Eukarya. Eukarya. Which are all what cell type? Eukaryotes. You can kind of guess where the names came from, right? So when we're talking about classification, right, if we just look at traits, we have to be kind of careful because you can kind of get this blurred line thing, and right? So my example here is whales, sharks, and fishes. You have a question, Tyler?
I was about to ask the definition of domains.
We're going to get to it. So a mnemonic that I learned for classification schemes when I was in school was, Kevin, please close our front gate slowly, right? So kingdom, phylum, class, order, family, gate, genus, and species, right? But we'd have to kind of augment that now because remember, domains comes above kingdoms. So I just changed my sentence to, damn, Kevin, close our front gate slowly, right? Another colleague of mine learned King Philip came over from Germany, right? These are play on words, right, that help people in remembering things. So this is mnemonic with a M, silent M, as opposed to, don't confuse it with something like pneumonic plague, right, which is with a P and refers to a pneumonia caused by a bacteria, right, highly infectious. And still occurs today, right? Plague still happens today, just not as widespread as it did in the past, because one of the major problems with that was sanitation. Right, and the fact that mice and their fleas carry this bacteria. None of us are cohabitating with those types of things by choice anymore, right? So taxonomy, as I said, that's that classification of organisms into categories. Phylogeny, on the other hand, is the study of the evolutionary development of a species, right? So what were the ancestors? What gave rise to the animals that you see today, right? So they believe that we were derived from apes, right? It was very closely related to them. Ontology is the study of the development of an organism, right? So how many of you guys knew we had tails, right? In our development process, we have a tail, right? But it doesn't continue to develop, right? It actually digresses during our developmental phase. But other animals that fall into the same grouping as us, right, like cats and dogs, right, other mammals, do have tails, right? Evolutionary-wise, we probably didn't need it, right? We're bipedal, we walk on 2 legs, we don't walk down on 4 legs. But then again, so do some apes, but they still have tails, right? They use it for other things, right? So it wasn't advantageous for us, so probably eventually phased out over time. So there is this possibility of this, our development kind of gives us a clue into our ancestry, right? The fact that we do have a tail during development, right? And that helps link us to monkeys and apes and things like that do have that. So this may or may not be true, right? That ontology replicates phylogeny or phylogeny replicates ontology.
What do you mean in our development?
So when you're in utero, in your mommy's womb, at one point you have a tail.
If there's just wagging around, just chilling.
It's very small, but you have a tail. And it digresses as you develop. So there are some people it doesn't happen, and they're actually born with a tail.
Yeah.
It's very rare, but yeah, it's true. So back to whales, sharks, and fish. So if you were to just look at, if we were characterizing all these guys, they do have the commonality that they all live in water and have fins and can swim. But are they quite different from each other? Yeah, we know that whales, for instance, are what we call mammals, right? They have hair. They give live birth. They have to breathe air from the surface. They have to surface to get air. Fish, on the other hand, have scales, right? No hair. They lay eggs, and they can acquire oxygen through their gills in the water, right? They do not have to surface to acquire oxygen for respiration. So their environmental pressure of an aqueous environment is probably why they all have fins, right? But many of the other differences tell us that they probably don't have common ancestors, right? They derive from completely different animals, right? Or they may have had a common ancestor and they diverged, right? But there's a whole bunch of beliefs as to, right, how modern animals came to be, and modern organisms, for that matter. So phylogeny is trying to reconstruct, right, that family tree that we don't know, right? Unlike this family tree, which is published on the internet, right, we know, right, they were able to figure out that these two guys, right, who their parents were and who their parents were, and extrapolate back to this common ancestor, right? The one that gave, if this guy didn't exist, these two third cousins down here wouldn't exist either, right? So what came before us, right? Was it some type of monkey, right, that eventually over time? And it makes sense, you know, when you look at the similar characteristics, too. And then we can even look at DNA, right? Right down to the cellular level, the similarities between us and them. And the very few differences that do exist that give us our different characteristics from them. So life forms are interrelated even, right? So a whole lot of crossbreeding going on. So probably one of the most supported theories of this is the endosymbiotic theory. And this is the belief that organelles like the chloroplast and the mitochondria were once in fact prokaryotes free living on their own and were engulfed by an earlier eukaryote. When this happened, of course, the eukaryote, that chloroplast, was a photosynthetic organism living all by itself. Probably what gave rise to the modern day cyanobacteria and algae, right? Those organisms, cyanobacteria, are just like a chloroplast. The difference is with a chloroplast, when it became engulfed in this symbiotic relationship happened between these two organisms, they lost the ability to exist without each other, right? So the chloroplast can't leave and live on its own anymore. The eukaryotic cell, the plant cell, the algal cell, right, cannot exist anymore without its chloroplast. Because it relies on that chloroplast to do what? Energy production from harvesting what? Light energy. Right? Because of the pigments and the structures. How do we know that this may be the case? Because chloroplasts have their very own DNA. They have their own ribosomes which make proteins. And guess what those ribosomes look like? Prokaryotic ribosomes. They're not eukaryotic. They're prokaryotic ribosomes. Where'd they come from? Well, probably a prokaryote. Right? Like the modern day cyanobacteria that we see. They lost the ability, though, to do everything on their own. Right? And again, that cell itself cannot survive without the chloroplast. Same is true for mitochondria. Mitochondria were once some type of bacteria. Probably the ancestors to the proteobacteria. Our cells cannot exist without our mitochondria. Right? They, again, are energy. Different way, though. It's not light energy. It's chemical energy. You actually get 99.9% of your mitochondria from your mom, right? The egg contains all the mitochondria. When the sperm comes, it's just basically mobile DNA. It has a little bit of mitochondria that it uses to be able to swim with its flagella, but all your mitochondria come from your mom. So they're all maternal. This also factors into genetic disease, right? If you get defective mitochondria from your mom, that's all you've got, right? You don't have that ones you got from dad to rescue you, unfortunately. So there's some mitochondrial diseases that are inherited maternally from your mom only because of that. A friend of mine in college was going blind because of that. He got defective mitochondria from his mom. And his sister, because of that, chose not to have children because she would pass on that trait to her children. And for some reason, it tends to manifest more in the males than females, which kind of sucks, right? For any babies. So she just has cats. She went that route. So does this make sense to you guys? All right, it's kind of cool, right? And we're going to talk about how we believe that even it went one step further. Later on, we'll talk about a second endosymbiotic event where an algal cell was engulfed by another cell, right? And that algal cell kind of deteriorated down into a chloroplast, or what's sometimes referred to as a plastid. So this domain system came to being when Carl Woese and George Fox, George tends to get forgotten about, the second guy on the list tends to get forgotten about. Back in the 1977, right, he proposed this, and based on his studies on ribosomal RNA. So remember, this is the RNA associated with your ribosome. All living cells have to have ribosomal RNA to make proteins. So when he did this, he saw this clustering effect. He saw all the bacteria clumping together. He saw all the archaea clumping together. All the eukaryotes and all the archaea. And the archaea at one time were kind of lumped in with bacteria in a kingdom classification that's not used quite as much anymore. You may have heard of Monera, right? So they actually found that even though these are both prokaryotes, right, they both don't have a nucleus, archaea, and bacteria, they're actually quite distinct from each other. Very different organisms. So much so they got their own classification, they got their own domain, right? So, as I said, in this course, we're going to focus on bacteria after this lab. But there are also other prokaryotes like archaea. And then today, we'll actually dive into eukaryotes, right, and eukarya. So where are humans? Eukarya, right? And we would fall under animals, right? We kind of put ourselves on this pedestal and we forget we are animals, y'all. We really are. Some of us more so than others, right? So by no means do I want you guys memorizing this next classification, and this classification would fall under domain. So we're in eukarya, and then we would have under that, and kind of even substituted or even above kingdoms. They're trying to decide, like, we don't have enough kingdoms, right? Are we going to get rid of kingdom? Are we going to change it? So right now we have what's referred to as supergroups, right? And there are, depending on where in the literature, right, what you're following, 5 to 6 supergroups. In your book, they list 5 because they combine these two into Euglenozoans. And a lot of times, these are phonetically said, these names. They can be a bit of a tongue twister. A lot of times I cheat and go to the How Do You Say website. But again, I don't want you guys memorizing this because, as you can tell, it's in flux, right? This stuff is going to change. But understand the concept here, right? That we're grouping organisms. So as far as memorization for you guys, you're going to be memorizing the name of the individual organisms we're looking at so you can identify them under the microscope. And the different things we're asking you to identify, like the nucleus and the chloroplast and things like that. So, but we're going to go through each one of these supergroups and look at some representations, right? So we're going to bounce all over the tree. So where are humans, though, on this tree? Because there's no animals listed here.
It's only bacteria and archaea.
No, this is all eukaryotes, right? Bacteria and archaea are scrunched down here and here.
Oh,
So these are all eukaryotes. So she's like, yeah, we're closely related to fungi, so I'm thinking it's probably in this lobe of the tree. And you're right. Metrozoa is animals. Metrozoa is animals. So Berkeley has an amazing website that I have the link for you guys. Definitely check it out on all the cool stuff like ourselves. All the animals. Fuzzy and non-fuzzy animals. Huh? Oh yeah, Unicata, that supergroup, yes. That would be us. So when we talk about prokaryotes versus eukaryotes, as it relates to the domains, which two domains that we're not covering today contain prokaryotes? Bacteria and archaea. What one domain contains eukaryotes, which is the focus of this lab? Eukarya. That one's easy, right? So again, there's some flux here, right? The common kingdoms that most people accept are the kingdom animals, plants, protists, and fungi. And then some informal classifications that we look at is protists usually include the protozoa and algae. And when we're talking about fungi, we're talking about yeast and mold, right? Those are common names that we understand, classification names, groupings of these types of organisms. But again, we're going to look at each one of these supergroups. So again, this is a classification scheme. Right? Above or replacing kingdoms. So we're going to start out at Excavata. I'm getting better at saying these names. I'm not even going to get into the subgroup, right? But so our representative of this subgroup is this guy. And then this subgroup, this guy. And do you guys remember this guy from your homework? Who is he? I got a whole poster. Yeah, the paired nucleus, because he almost looks like he has eyes, huh? Yeah, this is Giardia. This one's Giardia. And this guy, anyone know this one? You might have seen it in biology classes. A lot of times swimming around in your pond water. Nope. Euglena. It's Euglena. So all of these animals, or organisms, you should say, in the supergroup Excavata have these common attributes. Again, remember we're grouping them based on attributes. So they're unicellular, they have a feeding groove, they have one or more flagella, and they typically have a life cycle that has a trophocyte and a cyst phase. Anyone know what a cyst is? It's resting stage. So what is the trophocyte? It's the feeding phase, right? The active, alive, out there feeding phase. The trophocyte is the cyst is the resting stage. So it's not actively metabolizing, it's not moving. It usually walls itself in, a cyst wall, right? And this protects it from the elements when food is scarce, when environmental conditions are harsh. It can still survive. So then when it's picked up, and a lot of parasitic organisms form cysts, right? So when then it's picked up by an organism from the environment, it's going to come out of that cyst, right? Become the active feeding phase again. And this is very true for Giardia. Giardia actually has these sucking discs that attach to you and suck your mucus from your intestinal lining.
The SCP that was transmitted by the site?
Oh, I'm going to get to it. I'm going to answer all the questions that are in your thing as we go. Right, so we have these 4 subgroups, and for each subgroup we have a representative organism to look at. The unfortunate thing is today we're not going to look at a single one of these under the microscope. I had some Giardia slides, but again it's a fecal sample, right? So it has a lot of crud on it. And there weren't a lot of really good, well-stained Giardia on the set of slides that we got in. And I haven't been able to acquire any better ones. So we might be phasing that slide out and replacing it with another one of these where I can get better slides of it. I'd really like to get Euglena. Those are easy to get good slides of. So we don't have any of these guys for you to look at today. So we're just going to go by pictures in your books. If you have to identify them, we would put a picture from your book instead of look at it under the microscope.
We were supposed to have all this done before we came in.
Yep.
Because I could have bought a lot of this.
It's in your lab manual. I did it Tuesday night.
Posted online.
Yeah.
Should all be in those.
Mm. Yeah. If you read through it, it's all there. I checked. I did my homework Tuesday night. Okay. So, this is Trichomoniasis valigenaris. As I said, we don't have any slides. It'd be better if we got to see the live organisms. It's really fun to watch these guys swim around. I'm sure you could search YouTube and watch them wiggle. They're pretty cool. So they have a nucleus, they have flagella, they have an undulating membrane. That's why I said they really move really cool. They're only known to have the trophocyte phase. They're not known to form cysts. They are fermenters, incapable of oxidative phosphorylation. That's what we do with our mitochondria, is oxidative phosphorylation to make ATP. They're reliant just on fermentation, which means they don't produce as much energy from each molecule of glucose that they break down. They have Euglenozoans. These actually produce hydrogen gas under anaerobic conditions. So these guys tend to be really smelly when they grow. They can infect, typically for women, the vagina. So they'll cause a sexually transmitted disease referred to as trichomoniasis. Not to be confused with other tricks, right? Like trichinosis, which is caused by a roundworm called trichinella, or trichuriosis, which is caused by a whipworm. Often, but not always, causes vulva vaginitis. Vulva is the lips before the vagina for women. This can get infected by this organism or the vagina itself. When you ever hear itis, they're referring to an inflammation, right? So when you have meningitis, it's actually an inflammation of the meninges that surround your brain. That's not telling you what causes it, right? And for meningitis, viruses can cause that inflammation. Bacterial infection, a viral infection. You got to be more specific. You can't just say meningitis, right? Just means your meninges are inflamed, which is bad news, right? Because they kind of protect your brain and spinal cord, and those are a little bit important to life. Make sense? Okay. So all those itises out there, right? A lot of times we get extra, more in-depth information. A lot of you guys are allied health, nursing majors. You want to know every little detail. So one of my other colleagues put every little detail in there for you guys. We're not going to test you to that level, right? You just need to know trichomoniasis, right? So can you see the 4 pairs of flagella in this picture? Right, the picture in your lab manual even sucks. Can you imagine how bad the slides are? They're not good. And I have multiple years of work under the microscope. So does one of my colleagues who studied parasitology in grad school. We even, one day for fun, raced to see who could find the Giardia and the Entamoeba histolytica on our slides. I won one, she won the other, and it still took us a little while. And we have years of experience. You guys, if you want a real challenge, I left 2 of those slides in the study lab. All these slides that you really need to look at are in the study lab as well, 2 trays. I left one of the trays, I left the Giardia and Entamoeba histolytica on there. If you really want a challenge, if you can find it, I'll be super impressed. Like, super impressed. So. No. This class is so straightforward, you guys don't need bonus points. No. Trust me. Just do your work, come to class, study, you'll be fine. You don't need bonus. I promise. If you are struggling, you come see me, and we'll get you there, right? All right, so here's a drawing that shows you the better details of these cute little guys, right? So there's a trophocyte. As you can imagine, even on the internet, it's hard to find good pictures. I got a few up on that poster that one of my students made. So the mitrosomes, non-functioning mitochondria, again, they just rely on glycolysis. They have the paired nuclei, which kind of look like eyes, which is cool. The flagella, which they actually have 4 pairs of, so 8 total. They have 2 medial bodies. They do form cysts, which the unfortunate thing here is they resist water treatment methods for the most part. So they can contaminate water supplies. Typically, it'll make you really sick sometimes for some individuals. This was known as beaver fever because it tends to happen more in stagnant water. And beavers, of course, dam up flowing water into small ponds and such, which creates that stagnant water. So this infection is the most common intestinal parasitic disease affecting humans. And again, for a lot of us, you may even be an aseptic carrier and don't even know it. You're not having any symptoms. Anthony von Leeuwenhoek, actually, the guy that first looked at microscopic organisms and wrote letters to the Royal Society in London and is credited with the first to observe, he was the first to also see Giardia because he looked at his own poop.
So how do you treat?
Different medications. Well, right. It's a good question. Typically, antibiotics are against bacteria, but what an antibiotic is a substance that inhibits microorganisms or kills them that's produced by a microorganism. But we have other drugs out there that are, may have been harvested in plants or made by a chemist and stuff that will affect eukaryotic type organisms. But good observation. So Euglena, right? As I said, these guys are kind of cool. They too are motile, right? They're green because they naturally are able to photosynthesize because they have a chloroplast. So because they can use light energy, they have an eye spot so they can swim towards the light and detect it. It helps them sense light. That's that little red dot there. And the green is the chloroplast. These guys are also what's referred to as mixotrophs. They can make their own carbon from carbon dioxide. That's autotrophic when light energy is available. When light isn't available, they're going to eat other organisms, right? They're going to be heterotrophic. They're going to eat organic sources of carbon to be able to maintain themselves. So they have more than one way to survive. So what structure gives them their photosynthetic ability? They have a chloroplast. And it actually takes up most of their cell usually. Yeah. So that's why they're green like that. So trypanosoma, right? These guys are pretty cool. They have one enormous mitochondria. What disease does this parasite cause? African sleeping sickness. And actually, trypanosoma is just a genus, and they didn't specify in your lab manual. Trypanosoma brucei with the subspecies Gambesi causes African sleeping sickness. There's Rodensia as well that causes African sleeping sickness. There's another Trypanosoma cruzi, which actually causes Chagas disease. But we're just going to hold you to what's in your lab manual, which is African sleeping sickness. It's in there. So our next supergroup contains things commonly referred to as green algae or chlorophytes. So this cute little guy, which we won't see in this great of a detail under our microscopes. These guys are really tiny. But again, notice the green, it has a chloroplast. It's also motile. You see the flagella? And then, of course, the eye spot so it can find the light. These guys form very large colonies of cells, right? And so what you're actually seeing here is a whole bunch of these little tiny guys forming this big ball. and then even smaller balls on the inside. These smaller colonies are referred to as daughter colonies because they originate from cells within the larger colony. And these are actually, you could say, granddaughters, because this one is a daughter of this colony, which is actually breaking up. Do you see the edge of it? So we'll be looking for these balls and their, under the microscope, these colonies and their daughter colonies within them. And these guys are stained today, so they're purple. But they're naturally green.
So you're going to see the balls, we're not going to see
Yeah, you're going to see this. You're not going to see the detailed. We don't have the magnification ability to go this small.
At the school, or like at night?
Well, there's pictures of it, so somebody saw it, right? Our microscopes, no, but other microscopes can get to that level of detail, yes. So actually, these are pictures from a website, a link that I have. You guys, a really cool one, where the guy really likes Volvox, and these guys are pretty cool, I think. They're actually linked to each other by cytoplasmic extensions, right? That's this line you see there. That's how they're able to keep that ball shape. And then chlorophytes, you've got good old Spirogyra. And this is the easiest one for students to remember because of the spiral-shaped chloroplasts that run through the filaments. So these guys form filaments, right? Not a colony, but cells linked end to end in long chains. So for chlorophytes, we've got Volvox. We don't, unfortunately, have Chlamydomonas. Next week, when they get the live specimens in biology, and I'll try to steal some before they contaminate them, usually they feed Chlamydomonas to the amoeba. So if you get that one and you see the little things swimming around outside the amoeba, that's Chlamydomonas. But we do have Volvox slides and Spirogyra. And so these are very close relatives to plants. They have cell walls made out of cellulose, very similar to plants. They're autotrophic. They have chloroplasts. So here's cute little Chlamydomonas. Again, we don't have slides of these guys. I might try and get us some, but they're tiny, tiny guys. There's a really cool song that this guy, Andy Irwin, does. And one of my colleagues found the YouTube video of him actually playing the song. It's kind of cute. I like it. Just for fun. We don't have time to play it, though. Alright. So, Volvox, again, prepared slides are going to look purple. You're going to see these balls. You're looking for this big colony with the daughter colonies on the inside. So, these can form, the whole colony could be all sperm that will break up and run to find the other colonies that may be egg colonies where they're all eggs. Or they can reproduce asexually. And when they do that, that's when you see these guys, because daughter colonies are the result of asexual reproduction. And I didn't steal the picture, but they form a zygote when the egg and the sperm come together, and they form this kind of cool structure. And you can see that on that website. There it is there, and I have the link under my links page in Canvas. So asexual. So for Spirogyra, right, filaments, as you can see, end to end, they're haploid, which means one set of chromosomes. What are we? Diploid, 2 sets of chromosomes. Where'd you get the 2? One from mom and one from dad, right? So these don't really have moms and dads, but they can sexually reproduce. So we got in some new slides that show conjugation, where the 2 of them come together, and the genetic information is going to come across, and it's going to form a spore. And then out of that spore will bust out more filaments. But they're going to be different from the original, right? Because they actually exchanged genetic information and formed a whole new organism. So you need to be able to identify the chloroplasts, right? They're in spirals, these green lines you see here. You need to be able to differentiate one cell from another. You can see its cell wall. And then also, of course, the nucleus, which is clearly seen in these slides. And then if you see ones like this, that's the conjugation. and the zygospore that forms. So then we get to the cute guys you're probably familiar with, right? Swimming around in pond water. Anyone know what that is? Paramecium. These are centaurs. These are cool. I wish we had them, but we don't. Beletinium coli. This one does form a cyst. It is pathogenic. And here's a trophocyte. We do have these guys. And then this other group contain the diatoms. These guys are sometimes affectionately referred to as nature's stained glass because of all the different colors and how their cell walls are shaped. They refract the light differently, and so it almost looks like pretty stained glass. And they're made up of the same substance that glass is made out of. Here's actually another organism similar to these guys actually eating some diatoms. Right? So he's actually ingesting them. So you see them outside and then inside the body. Can't remember the name of the organism that's eating them.
So is cyst used for eukaryotes and endospores for prokaryotes?
No, endospores for bacteria. I don't know of any archaea that form that. But yeah, a resting stage for bacteria would be an endospore. For any eukaryotes, it's typically referred to as a cyst. So this is a kind of cool aside about organisms that can bioluminesce. So I'm going to skip over that for time today. So this is that second symbiosis endosymbiosis that I spoke about earlier. Where, remember, the first one was a pre-algal cell could have ingested a cyanobacteria, which basically became its chloroplast, or sometimes referred just as a plastid. But then they believe an algal cell was engulfed by these cells and began a symbiotic relationship where this algal cell basically deteriorated into the plastid. And again, how do they think, what evidence do they have? Well, it's molecular evidence, right? There are components in this plastid that are more closely related to an algal cell than to the cyanobacteria, right? But also components of the cyanobacteria are there. Right, so they believe it went this route. Cyanobacteria engulfed by algal cell then engulfed by these guys. So in this group we have what's commonly referred to as ciliates. So all these guys, the paramecium, the centaur, and the beletinium coli, they all are covered in cilia. They all have an oral groove, and then within the oral groove they have a section, the cytosome, that's considered the mouth where the food comes in. Then we have a non-motile one. We have the Plasmodium falciparum. And this has quite a complicated life cycle. Anyone know what disease Plasmodium causes? Malaria. Yep, causes malaria. So then our other subgroup, this subgroup, typically they have a flagella, but diatoms is the representative that we have, which doesn't have the main characteristic. It doesn't have flagella, but is very common in the environment. In aquatic waters, diatoms are very common. So we have our paramecium. Our slides aren't this detailed. They aren't specially stained so you can see the cilia, unfortunately. So we'll just be looking for the nucleus, and usually just the macronucleus. There are some really cool YouTube videos out there, right, where you can see the cilia moving, you can see the oral groove, you can see the contractile vacuole. But again, for our prepared slides, we're really just going to be able to see the macronucleus. We might be able to see some of this cool stuff you can see on YouTube if we get some live specimens in next week. So I'm hoping. These guys we don't have at all, the centaurs. So another ciliate. They have this really cool trumpet shape. They can swim around or attach. They're really fun to watch. Their cilia up here is where their mouth groove is, right? And they bring the food in. These dots are their macronucleuses. They have multiple nuclei. And there's, you can see within the oral groove, that little cytosome, that white spot is where the food comes in. They're naturally green, so if you get a a live specimen, they're going to have a greenish colorization to them, but they're not photosynthetic. These guys we do have, and again, the characteristic giveaway for this organism is this unique shape to its nucleus, this kidney bean shaped nucleus. They are ciliates, and again, if you get it high enough magnification on the edge and you focus up and down, you'll see they're kind of fuzzy, and so you can kind of see the cilia on the outside, but not for sure. So I remember I said this one's pathogenic, so what symptoms would you have if you had an infection caused by this organism? Be something you would ingest. Diarrhea, right? Bloody mucoid diarrhea. This one will actually destroy your intestines. That's during the acute phase, an acute infection. You might end up with a chronic infection where it alternates between constipation and diarrhea. Or you can't go and then you can't stop going. So again, more fun facts that you don't necessarily have to know. So then we have our Plasmodium falciparum. There are actually 4 different species of Plasmodium that cause malaria. This is the worst one. This is the most prevalent. And part of its life cycle, it actually reproduces inside our red blood cells. So the slides that we have are blood smears. So you'll see red blood cells. And in this case, the stain they use stain the red blood cells as well, so they're not red. The slides that we have for you guys, the red blood cells are red. And the parasites are kind of blue or purplish. And they'll be either inside the red blood cells or outside of the red blood cells, because they bust out of the red blood cells as well. So as we said, this causes malaria. Plasmodium causes malaria. And the mosquito is the vector that transmits it between humans and animals. And important parts of the life cycle happen actually in the mosquito. Asexual reproduction happens in us. Sexual reproduction happens in the mosquito. So you'll see it has all different morphologies within the, depending on what part of the life cycle and what stage of the life cycle the different Plasmodium are. So you'll see all different really interesting morphologies. The good news for you guys is you don't have to identify the different types, right? We just wanted, you can see that it's a parasite and you see it and you know it's a Plasmodium. So, whole bunch of asides, right? Malaria isn't the only thing transmitted by mosquitoes that can make you sick, and thankfully not prevalent here, right? We don't have that protozoa, the Plasmodium, that causes malaria here. We certainly have the mosquitoes. And different mosquitoes carry different diseases too. This guy carries yellow fever and dengue virus. This guy carries West Nile, which we do have here, right? And at one time we did have yellow fever and malaria in Louisiana, but we were able to get rid of it. We don't have the wild animal population, and we had the ability to detect and eliminate the source so that it didn't perpetuate in the United States. Not so good for other parts of the world. And then, of course, don't forget about things like ticks, right? And definitely check out the poster on the door on how to properly remove those suckers. Usually it's contaminated water for ciliates. Yeah. Something you ingest. So typically water, but water that contaminated your food could also happen. So you ingest it, either in food or water. Most commonly a contaminated water source is the primary source. Okay. So as you can see, malaria is a problem mainly in Africa, right, but also in South America and parts of Asia, in the Middle East. So most of you guys are probably familiar with the genetic defect of sickle cell anemia. Is actually people with sickle cell are resistant to malaria because of the changes that happen in the red blood cells that make it hard for the Plasmodium to be able to infect them. So because of that, sickle cell is very common in the African American population because it is beneficial in that environment where malaria is very prevalent. So some really cool information, detailed information about that. So moving on to diatoms, right? So diatoms are as I said, commonly referred to as nature's stained glass. These are aquatic brown algae, probably evolved from red algae, so these guys are photosynthetic. They can be single-celled or colonial. They're autotrophs. They use carbon dioxide to form their carbon molecules. They lack flagella, unlike most of the ones in this group. The cells have two halves that overlap each other, almost like the petri dish lid in bottom. And they have a golden brown pigment. But again, some of the specimens we have been stained, right? So they're not natural. And they have symmetry to them. Some of them are centric, where they're kind of circular or round. And then you have the pinnate, which are the more elongated forms. So you need to be able to find both of those on your slides and identify them. What inorganic compounding is incorporated into their cell walls?
Silicon?
Silica or silicon. Or silicon dioxide, actually, is the proper chemical name. This is the same thing that glass is made of, hence why these guys tend to reflect light and can be really beautiful under certain microscopic conditions where they look like stained glass. So Euglenozoans, right? So this is the entamoeba. These guys don't have a characteristic shape to them, right? They're constantly changing. How parasitologists would recognize them is by their very unique characteristic of their nucleus. It has this bullseye appearance, right? Where you have a dot and a circle around the edge. Here's cute old amoeba. It's actually getting ready to eat something, right? Using its cytoplasmic extensions. This is actually a drawing I found on the internet, but I thought it was so cool, right? Almost looks real. But this is an amoeba eating a paramecium. It's one of their favorite foods. So, our slides are not stained this well. They're all one color, but you can differentiate the nucleus. You do need to look for those cytoplasmic extensions. And again, they're dead, so they're stuck. But hopefully we'll get some live guys in. This is actually YouTube I took in this class of a real live one. And I have the links for you guys. And so you can see this guy change and move throughout the video. And then again, the little dot swimming around. Do you guys remember what organism I said that was? Chlamydomonas. If you watch that video of him singing, you'll never forget. Okay. So the species name actually comes from Proteus, which was a Greek god of mythology that ruled the sea, or the waters, rivers mainly. So these guys are found in fresh water. They're free-living. They have a nucleus, no cell wall, right, because they're constantly changing shape. And they do that by pseudopods, what's commonly referred to as false feet, these cytoplasmic extensions. These guys move pretty fast. It's pretty amazing how quick they move. So again, I have the YouTube videos of movement and feeding for you guys to check out. This is the one I said we don't have any slides, Entamoeba histolytica. We have found them. It's not easy. It would take you probably a couple of days, the skill level you guys are at. But you can find that nucleus. Here, too, they've also ingested red blood cells. This particular amoeba, the entamoeba, are parasitic. So they do have cysts as part of their life cycle. But again, what's really going to help you in identifying is that bullseye characteristic of their nucleus. So what sample would we use to diagnose? Again, you would ingest this, so it ends up in your stool, your feces. We take a fecal or stool sample in your poo. Which also makes it hard to find them on the slide because, again, there's usually a lot of other crud on the slide, right? So it makes it difficult to find the actual organism you're looking for. So a little bit of information about the infection in general. Then finally, fungi, right? We leave the fun guys for last. Yeah, I know. My humor's bad. Okay, so yeasts. right? These are unicellular fungi. They typically reproduce asexually by budding, and that's what you see here, are smaller. They don't divide evenly, right? Little buds come off of them. So you're definitely going to look for this under the microscope. Sometimes those buds will elongate and almost look like hyphae, which are a characteristic of molds. These aren't true hyphae, so they're typically called pseudohyphae. So when we talk about fungi, we tend to unofficially group them as yeasts and molds. So fungi in general, non-modal. They have cell walls made of chitin. This is the same substance that's found in the exoskeleton of things like crawfish and roaches and things like that, insects. Chitin. But it's spelled chitin. It's said chitin. But we're not going to argue tomato, tomato. It doesn't matter, whatever works for you. Okay, so the really interesting thing about these organisms, they're absorbent, which means they release enzymes into your food and then digest it and soak it up. So this is why when your bread gets moldy, it gets mushy, because it's digesting it. Your cheese, right? You can't cut the mold off, y'all. It grows into the cheese. Same thing with the bread. It grows into it. So that slide I have over there, it grew into the agar, right? If you were to look at those agar plates, it grows up out of it. Those are typically the reproductive structures with the spores that will release if we open those lids, which we won't, right? But it grows into, right? The filaments grow into the food. They release the enzymes, right? Digest the food and absorb it. So yeast are unicellular. They're vegetative, actively metabolizing, and then they can also reproduce by that asexual budding, which is that uneven cellular division that you need to look for today, right? So like that. So this was the first bud, right, getting ready to detach, and then another bud is starting right away. So that one's reproducing. Or again, these buds will start to elongate for some species under certain conditions. And you'll see this hyphal form, which is actually not truly hyphae. It's what we call pseudohyphae, false hyphae. Molds are multicellular filamentous. So these are the ones that actually form the long filaments that's called hyphae. Hypha, if you're just looking at one filament. If you're looking at multiple filaments, it's hyphae. And if you're looking at a huge mass that you can see with the naked eye, like on those plates or on your bread, it's referred to as a mycelium, right? The hyphae you can only see microscopically. I don't think any of you guys have eyes that can see that detailed, right? So only under the microscope do you see the actual hyphae, those individual filaments. What you see on a plate, right, is what we refer to as the mycelium. So classification is based on sexual spore production and on the presence of cross walls in the hyphae. And this is typically refers mostly to molds. For our yeast, we have two representatives, Saccharomyces cerevisiae and Candida albicans. For the molds, we have three species, Rhizopus, Aspergillus, and Penicillium. So for Saccharomyces, I think the picture in your book looks kind of like this. And the one for Candida kind of looks like this. But actually our slides look more like what they say for Candida. And, you know, something more like this. So they're going to be the purple dots. And sometimes you'll see the little nuclei inside. And again, you want to scan around and you want to try and find ones that are budding, you know, like you see here. Does this yeast cause disease in humans? Saccharomyces? No, we like this one because what does it help us make?
Let's make bread.
Bread, beer, wine, cheese, right? The good stuff. Baker's or brewer's yeast is this yeast, Saccharomyces. Unlike Candida, albicans, and this one under different environmental conditions, you'll see the mostly yeast form with the budding, but at body temperature, at 37, it'll form those pseudohyphae. So I have two slides. One are old slides, which actually contain those pseudohyphae. I had ordered some new slides, and unfortunately, even though I ordered the exact same slide, I guess they don't keep the conditions the same. I didn't say any pseudohyphae on the new slides. The new slides, you just see the you just see the budding, the yeast form. You don't see the elongation of the buds that pseudohyphae form. So make sure you look at both slides and try and find the pseudohyphae on the older slides. They're labeled, and they're the ones with the circular cover slip. So is this organism an opportunistic pathogen?
Yes.
Yes. This is the one that when women say I have a yeast infection, this is what they're talking about. And it's not necessarily you have an infection. We all usually have this yeast, right? It's just that under certain conditions it will overgrow, right? And there's lots of different conditions that can cause the overgrowth. And although it mainly happens for women, again in the vagina and vulva region, it can also happen in the mouth. It can also happen in skin folds for obese individuals because of the moisture, right, and warmth in that region. And again, the same problem for women, the whole moisture problem and warmth, right, when it comes to the vagina. And then, of course, the mouth is always moist, too, and warm. And that's what they like. So certain individuals are more apt to have these type of infections than others, and certain conditions will lead to having a yeast infection. If you're one of those lucky women sitting out there that have never had it, you really are lucky because it's very common to happen. So lots of fun facts. Brings us finally to the molds, right? That's a nice, pretty, hairy one. So again, we're going to be looking at their reproductive structures, right? When they go aerial and they form these structures that have the spores that are going to release into the air and hopefully land on something else. So they're going to have different structures to them, their reproductive structures. They're going to help you tell one species from another. And for one of the species, we actually have the sexual form. So we have Rhizopus, Aspergillus, and Penicillium. So again, when you see the mass on the petri dish or on your bread, that's a mycelium. When you look at it microscopically and you see the individual lines, right, that's the hyphae. And for this older slide I had, you could actually see this was the part in the agar where it was growing, and this was the part above the agar where it was growing up in the air, right, this fuzzy up part. But it was also in the agar too. You can see right here, that's in the agar, right? So for Aspergillus, I ordered us some new expensive slides too, because this picture in your book is actually a cross-section of this sexual of this reproductive structure. So I ordered these, and so you can actually see when they do it in cross-section, because there it's just like a big ball, right, covered in spores. But when you look at it in cross-section, you can see the spores line up, and that line of spores is called conidia. So you're looking for the hyphae, right, that'll be down the threads, right? The sporangium, which is this structure that holds the spores, right? And if you're looking in the cross-section one, the conidia are the line of spores you see here on this structure. So commercial available products that are generated by this organism. So we can ferment soybeans with 2 different species of Aspergillus and make soy paste and soy sauce. And citric acid is produced by Aspergillus nigra, commonly known as vitamin C. So actually really potent source of vitamin C. No, it's natural, right? It makes citric acid, and we can harvest it from it. That is totally natural.
Rasmus, what was found on food?
We haven't gotten to it yet.
How you had soy paste and soy sauce from soybeans, and then the white crop running responsible?
No. You don't need to know what species. You just need to know the products, right? So soy sauce, soy paste, and citric acid are made from Aspergillus, or made by. Citric acid is made by Aspergillus, right? We turn soybeans into soy paste and soy sauce using Aspergillus species. Right, yeah. Any one of the 3 products is acceptable, yes. So for penicillium, and we most famously know this mold by a product it produces that we steal all the time called penicillin, right? But this guy also does other cool stuff. And again, to identify it under the microscope, you're going to look at its conidia. And in this case, you can see it without it even being cross-sectioned. They form these long chains, again, the conidia, but it's just spread out like a brush. And actually, penicillium means paintbrush. That's actually how it gets its name, because its reproductive structure looks like a paintbrush. I think it looks like a broom, but, you know. So, what food, though, utilizes this mold? Cheese. The different blue vein cheeses, that's this mold, penicillin growing in it. So those of you guys with penicillin allergies are freaking out right now, right? You're like, oh my god, I can never eat blue cheese again. No. Under those conditions, usually in the cheese, it's not stressed. It's not fighting with bacteria for that food source, so it's not producing penicillin, right? Penicillium doesn't always produce penicillin, right? Only under certain circumstances, when it's stressed, basically, and fighting. It's also used as a preservative for sausages and hams. Its growth inhibits the growth of bacteria. Rhizopus is our last one, and this guy is found where, typically? On your bread. So we have 2 slides for this one as well. We have the asexual spores, and you have to scan around because there might be some sexual spores on there too, right? It's kind of hard to separate these guys. But this one's a little bit different. It has like this structure, and it's like a little cap, right? And all the little tiny spores are in here. So the cap busts open and all the spores are released, right? So you're looking for that. And then sexual reproduction, when 2 hyphae that can mate come together. So you notice this is the deteriorated hyphae. They'll come together and form this special spore called a zygospore. And that's a result of sexual reproduction. This will eventually form a sporangia, which will be, so here's its life cycle. So here it comes together, right? Forms that zygospore. And then it's going to form another sporangium that's going to release spores, right? And then the cycle starts all over again, right? They can asexually reproduce, or they could then 2 of them come together and reproduce sexually, right? So they have a choice. So that's one of the funny things in the Chlamydomonas video. He's like, we can reproduce sexually, or if we don't want to, we can just split, right? Asexually. It's a cute, short little video. I highly recommend it. So we've talked about a lot of pathogens. I know a lot of you guys are really interested in pathogenic organisms. So to help summarize the pathogenic ones we talked about, Peter Hickman put together a nice table that summarizes all the key attributes of the pathogenic ones to help you guys out. So that, too, I've posted on my Canvas site. Under this module, and I'll send it to Andrew because I think I forgot, so, and Sarah, so they'll have it. So it's right here, summary of eukaryotic pathogens. These pictures, right, today's lecture that I just did, and then later this recording will be right there.